Okay, guys. Happy Sabbath. I'm going to I'm doing this video on the 6th of June. I think I'll post it tonight. I don't know. But I want you guys to go over this with me. Okay, learn as much as you can. I'm going to send you some notes on nutrition. I want you to read that as well. And I want you to complete the test so we can do two honors to make up for last week. Okay, so if we finish these two honors, we'll have four honors. We only have two more to do. It can be done next week, and we'll be done with our honors. And when club opens back up, all we have to do is our book work. Okay, which should be simple enough. Now, what I'm going to do first is outline what happens in each organ. Okay, and then we'll, I guess, overview the process, and then we'll go into the actual requirements for the honor. Once we go on for that, there's a little bit that we will not cover that we'll just read over in the requirements, and that'll be it. Okay, no problem. Now, so in the mouth, in the mouth, mastication takes place. Mastication is the mechanical digestion of the food you eat. So let's say you ate a piece of bread with chicken in it, right? Um, the chewing that you do is called mastication. So that's mechanical digestion. Chemical digestion also takes place in the mouth, and that is done by salivary amylase. Amylase will convert the, well, isn't convert. Salivary amylase catalyzes an enzyme, the hydrolysis of starch, which is in the bread that you eat, into maltose. Okay, and the mucus in your mouth helps the food stick together, and the tongue forms the bolus so that it goes. So when you swallow and it goes into your esophagus, um, yeah, it can pass through easily, no problem. Right, and go through down. Now, next after the mouth, okay, of course you see the salivary glands there. Saliva is what contains salivary amylase, the mucus, all that kind of stuff. So let's not go into that. From the mouth, there are two places if we can go: the trachea or the esophagus. There's a cartilaginous flap called the epiglottis, which blocks the trachea, the trachea, I should say, which is in front, and then so the food goes down the esophagus. And by peristalsis, which is the antagonistic contraction and relaxation of the longitudinal and circular muscles, food goes down the esophagus into the stomach. Now, in the stomach, mechanical digestion also takes place. And that is because the stomach is a muscle, as you must know, and so of course it's crushing the food, stuff like that. And now the chemical reaction that takes place in the stomach is by pepsin. Pepsin is an enzyme which helps catalyze the breakdown of the protein, let's say that's in the chicken, from the bread you ate, the chicken and bread you ate, into polypeptides. So what happens is that the protein in the chicken can be is sorry, the building block of the chicken in the bread and chicken is amino acids. So you have amino acids, many amino acids make a polypeptide, right? And then polypeptides form proteins. So first the proteins are broken down into polypeptides. Later on they'll be broken down into amino acids, which is what we want. We want the simplest building block of all the food that we eat: the carbohydrates, the fats, and the proteins. Okay? So in the stomach, pepsin catalyzes the breakdown of um, proteins into polypeptides. We also have hydrochloric acid in the stomach, which kills any bacteria that might be on the food and stuff like that. And then we also have mucus in there. Okay, now all this together forms gastric juice, and the product of the mechanical and chemical digestion that takes place in the stomach is called chyme. And chyme is released by the pyloric sphincter right into the duodenum. Duodenum is the first part of the small intestine. Okay, now when the chyme enters the duodenum, it has, like I said, the gastric juice, which contains pepsin, hydrochloric acid, all that stuff. But the enzymes which work in the small intestine have to work at a high pH. They work best at a high pH. Enzymes work on two principles: sensitivity and specificity. I did not pronounce that word right. But what you have to know is that they are sensitive to two things: temperature, and pH. Since your body is kept at a constant temperature thanks to all the processes that your body does to maintain homeostasis, the one we are looking at right now is pH. In the stomach, the pH is 2 because hydrochloric acid is a strong acid, so the pH is really low. But in the small intestine, so pepsin works better at low pH, right? 2, that's given. But the enzymes I'm about to introduce in the small intestine work best at a high pH, around 8 or 9. So what happens is that from the pancreas, right, I want you to welcome the pancreatic juices. And the pancreatic juices contain sodium bicarbonate, pancreatic amylase, pancreatic lipase, and trypsin. Right, clap, 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 coming from the back, and then coming from the gallbladder, okay, formed by the breaking down of hemoglobin from red blood cells. We have bile, which contains bile pigment and bile salts. Now, the pancreatic juices are transported by the pancreatic duct, right, and the bile transported by the bile duct, right, from the gallbladder. Now, bile is formed in the liver, but stored in the gallbladder. You have to remember that. And of course, the pancreas has an exocrine and endocrine function. We are talking about the endocrine function right now. Wait, sorry, exocrine. Because we're talking about hormones, right? So exocrine function of the pancreas. So now that's happening. All this stuff, these juices are mixing up in the duodenum with the chyme that comes from the stomach. So let's talk about the function of each thing we spoke about. So first, I want to talk about the sodium bicarbonate. The sodium bicarbonate neutralizes the hydrochloric acid that was, sorry, what was it? That came from the stomach. It neutralizes it so that, well, to increase the pH. But there is more sodium bicarbonate than there will be hydrochloric acid, of course, right? So that means that even after it's neutralized, there'll be more there, which will increase the pH to the 8 or 9 that we are looking for. That's optimum for the enzymes that I just spoke about, right? So our first enzyme, let's look at our first enzyme, pancreatic amylase. Pancreatic amylase also converts starch to maltose, like I was talking about in the mouth, about salivary amylase. Next enzyme we're going to talk about is the pancreatic lipase. Now, pancreatic lipase and bile work together. First, the bile salts. Okay, first I said in bile is bile salt and the bile pigment. The bile pigment has no real function, just there. But the bile salts is what you want to talk about. So the bile salts break down the large fat droplets from, let's say, the chicken, we have a lot of fat, into smaller fat droplets that pancreatic lipase can work on. Right? By breaking down into smaller particles, there's more surface area for pancreatic lipase to come along and break it down into the fatty acids and glycerol, which is the smallest building block of the fats and what we want. The same way I was saying, we want the amino acids. Now, for the proteins, we have our trypsin. Trypsin continues the work of what happens in the stomach, breaking down your proteins into polypeptide chains. Okay? Now, so all that has happened. Do I want to describe anything else that's happened here? Well, I want you to remember that I think bile is a yellowish green watery substance. Right, should remember that. That might come in the test. Yes, so we're in the small intestine. So once that happens, I want you to think about, I want to tell you, sorry, that the small intestine is the end of digestion. That's the last place where everything is broken down. And it's where the carbohydrates, the fats, and the proteins are absorbed. So to this point, we've broken down only our fats to their basic unit, the fatty acids and glycerol. So we still need to break down our polypeptide chains into amino acids, and we still need to break down our maltose and other disaccharides into monosaccharides. Hmm? There are something called villi, which line the wall of the small intestine. and they are like little tentacles and stuff like that. That's the best way I can describe it. But around them, there are even more enzymes, cells that produce even more enzymes. And these enzymes, there is proteases, and different enzymes that break down, what do you call that, polypeptide chains into the amino acids that we want. And then we have maltase, which breaks down maltose into glucose. We have lactase, which will break down lactose into um, glucose and galactose. And then we have sucrase, will break down sucrose into glucose and fructose. So glucose, fructose, and galactose are all monosaccharides. So that's done. We have all our monosaccharides, we have our amino acids, and we have our fatty acids and glycerol. What's going to happen next? All these things are going to be absorbed by the villi into the bloodstream. Okay, so by active transport, active transport, diffusion, all these things are going to move into the villi into our bloodstream. Now, the fat is circulated by the lymphatic system, okay, which is a whole other story. But what we are looking at right now is our protein, our amino acids, sorry, and our monosaccharides. So they are transported by the hepatic portal vein to the big boy up here, the liver. And in the liver, assimilation takes place. And assimilation is basically how these things are how these basic units that we got by digestion are used. So for glucose, most of these are used for respiration in cells and stuff like that. If not, it's stored as glycogen in the cells of the liver, or it will be stored as fats, as adipose tissue under the skin. Okay, our amino acids will be used to produce stuff like hormones, antibodies, enzymes, mm, muscle tissues, all that kind of stuff. And if it's not used for that, excess of them will go through deamination. And deamination is basically the process by which the amino acids are broken down. Amino acids are broken down into ammonia and I think a keto acid, I believe, and then that ammonia reacts with carbon dioxide to form urea. Now, urea is excretory product that will be released by the kidney later on and like that, but I wanted to give it a full idea. You don't have to know this for this one specifically, but it might come on test, so it's fun to know. So next, I said that was we were looking at the fat, but let's look at the fat now. So the fat will be used to produce the cell membranes of cells and stuff like that, and it will also be, like I said, stored under the skin as adipose tissue. Now, what I did not mention when I brought our monosaccharides into the liver was that your body works in glucose. Glucose is what it needs, glucose is what it wants. So the lactose and the fructose, galactose that we have there, these two other monosaccharides, the body doesn't want that. So the cells in the liver will convert the galactose and fructose into glucose. So everything is glucose. So now all our monosaccharide is glucose. And then this glucose will be used for respiration or whatever, or used as an energy store or whatever. Okay? That's everything I have to say about the small intestine. That's what happens there. We've spoken about assimilation. So what's not absorbed by the small intestine is released as feces, of course. So it goes through the small intestine, it goes into the large intestine. And in the large intestine, water is absorbed from the feces, minerals and vitamins are absorbed, and then it's held in the rectum until it's time to, be released when you go to the bathroom. But the longer it stays in there, the more water is absorbed. And that's why they tell you when you feel to go use the bathroom, you use it. But it stays there for too long, The large intestine will absorb as much water as it can from the thing, and you will become constipated when the stool becomes hardened. Okay? Anything fun you should know? I think that is it. So the overview. Your food comes from your mouth, goes into the esophagus. Of course, digestion occurs here. No digestion occurs in the esophagus. Digestion occurs in the stomach, both mechanical and chemical. The liver, gallbladder, and pancreas are known as accessory organs because they just provide stuff, but no digestion actually occurs there. The small intestine does more than you think it did, right? Large intestine, well, it's called that because it's larger, not because it's longer. The small intestine is the longest one, but it's actually larger, and the small intestine is actually slimmer, more of a narrow tube. Okay, the small intestine has a very large surface area which is created by the villi for the absorption of food. That's what you have to know. Okay, all right. So we are continuing the questions, criminalization, honor. Where does saliva come from, and what are the three functions of saliva? Saliva, often informally known as spit, is a clear, clear, moist, and usually somewhat frothy substance produced in the mouth. Saliva is produced in and secreted from the salivary glands, which we saw in our diagram before. Saliva moistens food so it can be swallowed easily. It contains an enzyme which breaks down starch into maltose, and saliva protects teeth from decay by plaque. Saliva is used to neutralize acids made from sugars in the mouth, therefore helping to prevent demineralization. Remineralization is when saliva helps repair the damaged crystals of the tooth enamel. Okay, so what happens is that there is a bacteria, can't remember the name of my head, but it basically metabolizes the sugars on your teeth, and then it some how do I say that? Excretes acidic substance, and that is what eats away at your enamel and causes decay of your teeth and stuff like that. So that's why I recommend you brush your teeth. The toothpaste contains something that neutralizes the acids that the bacteria produces, and and protect your teeth. But the enamel is made up of the hardest substance on earth. Hardest substance the human body produces, which is calcium phosphate. And that is what they're saying here. And potassium phosphate is an ionic solid, right? So that's what I guess that's what they're referring to here when you talk about remineralization and demineralization. Okay, that's not something we have to go into. The dentine, this thing is between the enamel and the pulp cavity. The pulp cavity contains nerves and blood vessels. That's where you feel pain if the decay goes deep enough. You feel pain if a wind blows, or if you drink cold water or hot tea or whatever, you feel pain. Then you have the gums, which I'm sure you can see when you look in the mirror. You have the cementum. We have the periodontal membrane. Okay, you can read that. We have gone over the entire system. There's nothing here that you don't know already. We've gone over chyme, we've gone over bolus, we've gone over where bile comes from, where it's stored. We're talking about the villi, talking about water here, fiber. Okay, so basically what you have to know here is that if you have a lot of fiber in your diet, it can help you feel filled. Okay, so you eat less snacks and stuff like that in between breakfast, lunch, and dinner, which can help you lose weight and fight obesity. Is what they're saying here. It can also help the GI tract to move food along smoother. Okay, it's like roughage, like they say. Okay, so it cleans out and it passes through. We've spoken about. I will link this in the description. It's a test used to test for like saliva and starch in food and stuff like that. Where you put iodine, it reacts with if there's starch, it reacts with the starch and forms a bluish black color. So you can easily be identified. It's normally used to test for starch in leaves, but it's also used in food test. It's called a food test. We've spoken about where these are absorbed, the monosaccharides, disaccharides, and polysaccharides. Amino acids are, like I said, acids. And acids have a very low pH. So you don't want too many acids or too much acid in your blood that your pH should be too low, and that will affect the enzymes and proteins all over your body, which may need a high what do you call that? A high pH to operate. Like I said, enzymes are very specific. I mean specific, sensitive. That's the word, sensitive. Specific is when you're talking about what substrate it reacts with. Like salivary amylase will only work for starch, and pepsin will only work for protein. Pepsin will not work for starch, and amylase will not work for a protein. Okay, that's what I mean when I say they are specific. What is ATP? ATP is basically an energy currency. Um, when you oxidize glucose using oxygen, you get ATP, which is used by your cells and stuff, life processes, for point of respiration. You don't need to know these processes above your pay grade. ADEC is your fat soluble vitamins, right? Water soluble vitamins are all your B's and your C. And then they want you to read these four texts here that refer to digestion. Digestion. You're going to do that on your own. Good luck preparing for the test, guys. I know you can do it. Have a good day.